Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to a special bonus episode of Prospective Doctors MCAT Basics. In this bonus episode, I'm going to talk a bit about coronaviruses, get into a little bit of virology, and then I have an interview with one of the members of the research team that developed the first coronavirus vaccine, and she's awesome and really smart, and I think you guys will find it pretty interesting. And of course, I doubt you'll see anything coronavirus specific on the MCAT, but I know you'll see some of the more general virus concepts on the MCAT, and so I hope that this podcast is kind of an interesting way to learn about viruses, vaccines, and you start learning about some topics in that area that maybe you hadn't heard about before. So before I throw you to the wolves and we start talking about this vaccine, I first kind of just want to introduce the family of viruses known as coronaviruses. So coronaviruses are enveloped, single-stranded, positive sense RNA viruses, and they contain the largest known RNA genomes of any virus. And to put this in perspective, the novel coronavirus that we're dealing with now has a genome that is about 30 kilobase pairs in length. In other words, it's made up of 30,000 RNA base pairs. Now you compare this to a virus like HIV, which has a genome length of about 9.2 kilobase pairs or 9,200 base pairs. Coronaviruses just have a much bigger genome. And when I talk about coronaviruses being positive sense RNA viruses, or I guess their genomes being positive sense RNA, this has some important implications. What essentially this means is that the RNA genomes that they have can act essentially as mRNA in the host cell and be directly translated into viral proteins by the host ribosomes. And this is in contrast to negative sense RNA viruses, where the RNA is complementary to the mRNA that is needed to produce these viral proteins. So in a negative sense RNA virus, this RNA genome first must be converted to its positive sense form by an RNA polymerase before it can be translated. In other words, as soon as positive-stranded RNA enters a cell, it can be directly translated into viral proteins. It doesn't need to undergo some intermediary step before that process can happen. And I will mention that this positive sense RNA that this coronavirus has does actually resemble a transcript quite a bit. It has a 5' cap as well as a 3' poly A tail. So it's got some of the features that, a transcript floating around in the cytoplasm of a cell has. And in the same way, it can be directly translated into protein by a ribosome. The novel coronavirus genome encodes 4 structural proteins and 6 accessory proteins. These structural proteins are proteins that show up in the mature, assemble the virus particles, whereas the accessory proteins don't actually show up in these mature viral particles. With that said, the structural proteins that the genome encodes are the spike protein or the S protein, the membrane protein or the M protein, the envelope protein or the E protein, and then the N protein. And I'll get more into these in the interview. On the other hand, these 6 accessory proteins are involved in influencing a wide variety of different cellular processes, including cell proliferation, programmed cell death, as well as cytokine production. And all of these accessory proteins seem to play a role in viral pathogenesis. So again, they're not actually included in the mature virions. So if you were to get infected with the novel coronavirus, right, the particles that were infecting your cells wouldn't contain these accessory proteins. It would contain RNA that encodes those proteins. But these proteins are more produced to affect the cells that are infected in effect than the viral pathogenesis. And the last thing I'll mention here before I get into the interview is that once we sequenced this novel coronavirus, which is also called SARS-CoV-2, we noticed that it had pretty high homology to a few SARS-like coronaviruses that come from bats. So for instance, if you were to take 2 novel coronaviruses from 2 individuals, say you just are sequencing the virus from two individuals, you'll see there's a sequence identity of around like 99 to 98%. And then say you also compare the novel coronavirus sequence to the SARS-CoV-1 sequence, and SARS-CoV-1 is just an earlier coronavirus outbreak that happened in the 2000s. You would see that those are only about 79% the same. Now, if you take the novel coronavirus and you compare it to several bat SARS-like coronavirus samples that were collected in eastern China, you can see about 88% homology. In other words, they share about 88% of their sequence. And so kind of the current working theory is that bats passed this on to humans, which then humans started transmitting from human to human, which caused this whole big pandemic. And there was some speculation as to whether there was some intermediate host that was facilitating the emergence of this virus in humans. They talked a bit about pangolins or pangolins. I'm not sure if I'm saying that right. It's not penguins. It's a whole different type of animal. If you are interested, just go Google pangolin. So people weren't sure if it was pangolins. Others suggested that it might be civets, which were responsible for the 2000 SARS-CoV-1 outbreak. So they don't really know if there's an intermediary host. I don't think there's been a consensus on that yet, but that's another part of the working theory that is something that could have happened. And right now they have also traced it back to the Wuhan seafood market. It looks like some of the first cases were from individuals who had been there. So the theory is that somehow the virus was either transmitted from bats to humans or from some intermediate host to humans and then spread.
My name is Olubukola Abiona, and I am a third-year post-baccalaureate research fellow.
Olu works in the Vaccine Research Center at the National Institutes of Health. She works on the coronavirus vaccines and immunogenesis team in the lab of Dr. Barney Graham. And I am lucky enough to be joined by her today to discuss the novel coronavirus vaccine. In general, a vaccine is a biological formulation that provides immunity to a particular disease. And typically a vaccine contains an agent or a protein or something that resembles the pathogen that is causing whatever disease that you're creating a vaccine for. Olu explained that when you give someone a vaccine,
the immune system produces a response where it recognizes it as a foreign particle, and then it makes antibodies against it.
And essentially, these antibodies that a vaccine elicits can then respond to and recognize very quickly the real pathogen if you were to get exposed. And therefore, you have a really quick immune response and you can kind of get rid of the pathogen very quickly and you don't end up getting really sick. And this vaccine that's being produced at the Vaccine Research Center is the first that actually made it into clinical trials. But there's a lot more vaccines that are all being produced by various research labs in the world.
Yeah, there's a ton of different vaccines that are being developed. I don't know if, and this is more like an offline thing, but there's like a chart. But basically, people are trying like RNA vaccine, they're trying protein. I think some people have tried inactivated viruses. Some people are thinking about virus-like particles. So there's like a ton of different efforts, and it's kind of great that we're able to all like concurrently fund or fuel each other's research that way. And I think that's really amazing from like a scientific point of view that like it's not just one effort, but multiple people coming together to fight like the common enemy, quote unquote, this SARS-CoV-2 virus.
And this vaccine that was produced at the Vaccine Research Center is actually an RNA vaccine, and this is a newer platform for a vaccine. So essentially what happens is a patient is administered RNA, and I'll kind of get into exactly how this works, but they're administered mRNA, and this mRNA encodes for a protein.
So it is coding for the spike protein.
So this mRNA that is delivered as a vaccine encodes for a particular form of this spike protein, and I'll get into more of what that form is, and Olu does a great job explaining that. But I first want to get into the functions of some of these structural proteins that make up the SARS-CoV-2 virus. And again, those proteins are the spike protein or the S protein, the M or membrane protein, the E or envelope protein, and then the N protein. Let's start with the spike protein, since this is the protein that is the antigen for this vaccine.
So, spike protein is basically it's mushroom shaped and it has two parts to it. So it has this head that kind of has like a three parts or three prongs to it. So the top part of it is what binds to its receptor, so the ACE2 receptor. And so what's really interesting about the spike protein is that it is in its pre-fusion conformation. Um, and it's a metastable kind of conformation where once the spike protein interacts with the receptor and binds, then it triggers a conformational change that's irreversible. That makes it flip into a post-fusion state. And that fusion is what allows the virus to basically enter the cell.
And there's actually two components to this spike protein.
Of it. So the S1 is the head that I was referring to. And the S2 is the stem region of the spike, or what's nearest to the membrane of the virus. And so that S1 binds the receptor, but when it binds the receptor, the S2 carries the fusion machinery that allows the virus or the spike to fuse to the membrane. So it's a fusion protein that's built into the spike itself.
So the spike protein is extremely important for this virus to get inside the cell, since it is the part of the virus that actually binds to the receptor on the cell in order to enter the cell. And this receptor that SARS-CoV-2 binds to is the ACE2 receptor, or the angiotensin converting enzyme 2. And it's an enzyme that is attached to the outer surface of many different types of cells in the lungs, in the arteries, in the heart, kidneys, intestines, and more. And also, if you've ever seen images of this virus, the spike protein is these large protrusions that are coming out of the virus's surface, and it gives it this really kind of almost pretty appearance where it looks like it's wearing a crown, and hence its name, corona, which is Latin for crown. The M protein, or the membrane protein, defines the shape of the viral envelope and is also involved in facilitating viral particle assembly. In the same way, the envelope, or E protein, is also involved in facilitating viral particle assembly, and it's actually required for the viral assembly. If you don't have it, these particles can't actually form. And then lastly, the N protein, or the nucleocapsid protein, is a protein that actually forms a complex with the genomic RNA of the virus. And it's important for also assembling these viral particles and getting the RNA genome inside of these particles. All right, so back to the vaccine. So again, the vaccine is an mRNA that actually codes for the spike protein, and actually a very specific form of the spike protein.
Is coding for the spike protein, but in particular, it's a pre-fusion stabilized protein.
I will mention that when she says stabilized pre-fusion protein, stabilized just refers to the fact that there's a few amino acids that are changed in order to keep that protein in that pre-fusion form and make sure it doesn't flip to the other form, this post-fusion form. In other words, it's stable in the pre-fusion form.
Stabilized protein, or version or conformation of the spike, and that's important because what we see in coronavirus research is that when you give the pre-fusion stabilized version versus like a wild type spike, which can flip into this post-fusion conformation or something like that, you tend to produce a more robust and more neutralizing antibody population than just giving like other parts of, or giving like a post-fusion spike or so.
In other words, the pre-fusion form of the spike protein actually elicits a better neutralizing antibody response than this post-fusion form of this protein. Because remember, again, once this spike protein actually binds to the receptor, it changes form. And if you're like me, you might be wondering, okay, what is a neutralizing antibody response? Why is that important for a vaccine?
So neutralizing antibodies are really important because they are highly researched to prevent viral infection.
So antibodies are little protein molecules that are secreted by B cells, and these antibodies are then able to go bind to virus particles, bacteria, whatever pathogen you might be exposed to. And once they bind, they can do a few different things, and you can go listen to the podcast I did on this. But Essentially, the antibody can either neutralize that pathogen and make it not infectious anymore, or it will bind to it and not neutralize. So you could imagine then that a neutralizing antibody is one that is actually preventing infection. And so that's what you want from your vaccine. You want to produce a lot of neutralizing antibodies. Back to Olu.
And how a vaccine produces it.
It being a neutralizing antibody response.
Is that basically we have like a structural or rational design where we take an antigen, we inject it into different animal models, and then we see the response. And then we can take the antibodies generated by that response and test the neutralization assay to see how potent it is. So by doing we get feedback on the antigen that we made, and then know, oh, is this a good vaccine candidate versus another formulator or different or antigen with different mutations that we might try out.
Another thing that I find really interesting is the theory behind why this pre-fusion form of the spike protein elicits a better neutralizing antibody response than the post-fusion form.
So when you think about it, and this is also seen in another virus in our lab that we study, the respiratory syncytial virus, when you give the pre-fusion conformation or pre-fusion stabilized protein, you have more epitopes that are exposed.
Epitope is another virology word, or I guess it's probably more of a biochemistry word, but it's the part of an antigen molecule to which the antibody itself binds. So this is the sites on the spike protein where the antibody would bind. So when Olu says that there's more epitopes on the pre-fusion form of this protein, that just means there's more sites for an antibody to bind.
You have more epitopes that are exposed that interact with the receptor more than when it flips into its post-fusion state, because essentially it's kind of like you have this open door when it's in pre-fusion state, and then when you give it to the body, it's like, okay, we can recognize different parts of the door, right? But when the door is closed, it's like it can only recognize like the frame of it or something like that, where it can't see the whole thing, but it can only see what's given. So by giving the pre-fusion, you have access to more epitopes that would generate more antibodies that would bind to the sites that the receptor will normally bind to, and that allows it to neutralize a lot better. It produces a more enriched or mixture of neutralizing antibody population.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
Another question that popped up into my mind was, okay, you have this vaccine that's an mRNA-based vaccine, and when you give it to cells, the cells are going to uptake that mRNA, and they're going to produce the spike protein inside the cell. Why not just express the spike protein in, say, like an E. coli, and then purify that, and then give the spike protein itself as the vaccine?
So some of the advantages, and one of the reasons I think that we went with this platform is that mRNA is cheaper and easier to produce. So if you want to formulate and mass produce it for, in this case, making a vaccine for the public, it is a quicker platform than, let's say, protein, because in that, even just a normal protein production of a spike can take 5 days to make, so like that. But giving an mRNA is more cost effective, because you also have to think about
when you're producing a vaccine, you also have to consider the company. In this case, Moderna is actually the company that's mass producing this vaccine.
Basically, it's cost of production is one of it. Another thing is when it comes to the protein itself, it could be not less stable, but you also have to have different formulations to keep it and do testing on that. So it's an extra step. And also it can also decrease the yields depending on how much protein you're able to produce per batch for a vaccine. And you also have to think about, like, if you're talking about, like, flu vaccines or things like that, how it's produced on mammalian cell lines or egg cells and how long that takes as well. So this allows you to skip or bypass that step and just directly give it to the body in a way that is not only cost effective, but easy to distribute.
So this vaccine was produced in record speed. It took something like 65 days to get it done, which is very, very, very fast. And Olu said that this is due to the fact that, first of all, it's an mRNA vaccine, as she just talked about. But she said there's more factors involved than just that.
So I think another factor is that for our lab, we've been studying coronaviruses for over 5+ years, and like even for that IPI, I've been studying coronaviruses in some capacity. And so we're really interested in pandemic preparedness. So we have been looking for generalizable ways to make coronavirus vaccines for different strains of it before this. And so from that previous research and from like the previous experiments, we're able to inform our current design. So we were able to kind of streamline that process because we know that generally certain aspects of coronaviruses are generalizable based on what we have researched before. And that was part of the reason we're able to streamline it so fast.
So it's not just that this lab was in the right place at the right time. They've been studying coronaviruses for a long time.
Lab has been studying human coronaviruses in general. So yes, the outbreak of SARS in 2002 did also spearhead our efforts. But also the outbreak of MERS in 2013 also contributed to that. And then you also have to think about, so there are 6 strains of coronaviruses, or 7 now, that can affect humans. And then when you talk about the 4 of them are endemic and cause the common cold. So in general, coronaviruses are known to make up about like 20 to 30% of common colds. So even if so 20 to 30% of the common cold. And so this mixture of like endemic and epidemic and pandemic coronaviruses really was an interest of our lab because it's like you can make a common cold vaccine or you can make one that's against like these more severe strains. And it's just a really cool aspect to think of because even if like you have an endemic or cold strain of coronavirus that gives you mild infection, you also have to think about the fact that like people have other coexisting conditions or have co-infections. So having a vaccine against that to mitigate any type of severity for another co-infection or comorbidity, a really great research area to target as well.
Another thing that's interesting to me about these RNA viruses, and remember, coronaviruses are positive strand RNA viruses, is that they tend to mutate quite quickly. And so this is just due to the fact that RNA polymerase, which is an enzyme needed for replicating the viral genome, tends to be fairly error-prone. In other words, when the RNA genomes are being replicated in order to be packed into these viral particles, there tends to be errors that are created during this process, which then causes the virus to mutate. And so, in general, RNA viruses tend to have fairly high mutation rates. And so because of this high mutation rate, one of the things you might worry about is that once you administer this vaccine, the virus is just going to mutate, which, by the way, is a random process. It's not like this virus is choosing to mutate to evade this vaccine. But the worry is that you administer the vaccine, and then the virus mutates, and that vaccine is no longer protective. However, there is a catch to this line of thinking when we're talking about coronaviruses.
I don't necessarily worry about that.
Say we compare coronavirus to influenza, which is another RNA virus that has a really high mutation rate.
The difference between coronaviruses is that coronaviruses have their own proofreading mechanism to basically limit mutagenesis or mutations. So while it is going to mutate, it's not necessarily going to mutate as fast.
As fast as influenza, of course.
And when we look at other strains of coronaviruses, we don't see that it necessarily mutates that fast as well.
So as you probably know, DNA polymerase has 3' to 5' proofreading activity, whereas RNA polymerase does not, which is why RNA viruses tend to undergo more mutations. However, Coronaviruses actually encode their very own 3' to 5' exoribonuclease protein that is independent of RNA polymerase. And as Olu said, this is a protein that is encoded for in the coronavirus genome itself, and it's also called NSP14 or non-structural protein 14. So overall, this is a protein that can go back and fix mistakes that were made by the RNA polymerase, which is actually really good for us as we try to create a vaccine for this virus, because once we create that vaccine, there's less of a worry that it's going to mutate and the vaccine is no longer going to be effective. So right now you may be thinking, okay, we've got this great vaccine candidate. It's an mRNA vaccine. It's going to produce this pre-fusion form of the spike protein. Why is this not being injected into people right now? I mean, we've heard over and over how it's going to take about a year to 8 months to create this vaccine. Why is that when we already have a vaccine that's made?
So I like to, and I've got this question a lot, where people were saying, oh, this company made a vaccine in 3 days. What people have to, or what's really important to realize is that we're making vaccine candidates, but it doesn't become an actual vaccine until it's tested in goes through phases of testing in animals and then human clinical trials. So even though this is a really dire situation, you don't want to get to a point where you skip those steps, right? And then you're giving it to people who have all these different genetic makeups, all these different health conditions and things like that. You skip the testing phase, and then it actually exacerbates the situation or isn't as effective as you was, right? So our goal and what we're trying to do is be as efficient and accurate as possible and the quickest timeline, but we're not skipping steps in this, right? Because those are really important.
For those of you who are not familiar with a clinical trial, for vaccines, there's 3 phases. So in phase 1, you start with 20 to 100 healthy volunteers, and you're just injecting the vaccine at different doses to see if it's safe. So you look for side effects that were unpredicted and maybe that didn't show up in the preclinical work. Then once you get past phase 1 and you see that it in fact is safe, now you do a pretty low-scale efficacy study where you inject several hundred volunteers with the vaccine and see if it works, right? One arm is a placebo, one arm is a vaccine. Essentially, they look to see if less people get the virus in the vaccine arm versus the control placebo arm. And then once that passes, they go on to phase 3 where they take thousands of volunteers and they do the same type of efficacy study in a bigger cohort. And as you could imagine, this is a pretty time-consuming process, these 3 phases. And so this is going to be the bulk of the time that's required to produce a vaccine for the novel coronavirus. And the phase 1 trial for this vaccine was started on March 16th, so about a month ago in Seattle. And I think they've expanded it a little bit, and now they have a cohort that is at the NIH in Bethesda, Maryland. But I kind of imagined that the hard part of this was over for Olu and her lab, and maybe they could just relax now, but that is not the case.
So typically you do have animal studies, so it typically starts in a mouse model, then moves up to larger animal models. So we're currently evaluating the responses that we've gotten from our animal testing and seeing the effectiveness of the vaccine in those models while we're also...
And they're doing all of this while the phase 1 trial is going on. Another thing that's really interesting to me about this virus is the fact that it can infect so many different species, right? We talked about bats, we've talked about civets, we've talked about pangolins, and then recently there was a story about this tiger, I think it was in the Bronx Zoo, that was diagnosed with COVID.
I mean, that's one of the things that I found really interesting about coronavirus is the broad tropism, as we were talking about.
Viral tropism is just defined as the ability of different viruses to infect different cell types or different tissue types. For instance, HIV binds to the CD4 receptor, which is mostly found on immune cells, specifically T helper cells. So you can say then that HIV has tropism for T helper cells. Back to Olu.
Here we are more focused on the human aspect of coronaviruses and health and disease, but we do consider okay, There's some strains that can infect pigs. This is coming from camels. Like, what is the relationship between that and how this virus is being transmitted, right? So a more epidemiological approach as well. So we do consider that when we are designing or thinking about like vaccines and also animal models as well.
And what about that poor tiger at the Bronx Zoo?
Yeah, that was very. I'm surprised by that. New strain, so you don't know how it's going to behave just yet, right? And the research is ever evolving. But I mean, there is another strain of coronavirus, not this one, that is like feline coronavirus that can infect cats. So I was like, it's not too far of a stretch if it has some sort of tropism in that. But we do see that this seems to have like a broader tropism compared to other strains.
A question that immediately popped into my mind was, okay, if this virus has such broad tropism and can infect a bunch of different animals, a bunch of different types of tissue, should we be worried that we're going to pass this virus to our dogs and maybe our dogs are going to pass it to us?
So I think, so I can't say whether or not you should be worried about that. I mean, there's no stories or research that have come out about that yet. I mean, there was like this one story, but that later on seemed to have gotten debunked about someone transmitting it to their dog. But it does seem like even if it was transmitted, it's not really necessarily being passed to humans from the dog. But that is still like, I can't say that for sure because I have to wait on more research on that. But people are looking into that, the effects of like pets, and if they can get it as well and transmit it to their owners or not.
And Olu brings up an important point as to what's kind of driving this broad tropism for the novel coronavirus. And it has to do with the fact that this virus can bind the ACE2 receptor of many different animals.
Yes, it can use the ACE2 receptor on all these animals, but the other thing you have to recognize is that not all ACE2 receptors have the same amino acid composition that allows it to enter. So, for example, it seems as though the ACE2 receptor in mice is different enough that it doesn't allow for infectivity. So it's not saying that, if there's ACE2 in all these species, it's going to affect all these species. The ACE2 receptor itself can be different enough that it doesn't infect all the species.
In other words, if an animal's ACE2 receptor looks enough like bats or enough like humans that this novel coronavirus is able to bind to that ACE2 receptor, then that animal is going to be susceptible to getting infected with this novel coronavirus. Olu, thank you for coming on the podcast. Obviously, I know you're busy, so thanks for taking the time to do this.
Sounds good. Thanks, Sam.
I have one last question for you, and that is, what have you learned throughout this whole process that is something that is kind of interested you and you wouldn't maybe have thought about before until you've seen it firsthand?
I think one of the really cool parts from like a research perspective about making this vaccine or just making vaccines in general is that it's really a multi-dimensional process. So it doesn't span just in like research field or medicine, but it goes like even further to like global economics and politics and public health, and it just reaches all parts and facets of life. And I find that like really cool how interconnected. Like the one thing that I'm doing in the lab that's like really tiny but basic can have an impact on so many levels and involves so many parts and people in the process as well.
And that is it for this bonus episode of Prospective Doctors MCAT Basics. As always, thanks for listening, and thanks a bunch to Olu for coming on the podcast. I think it was really interesting. I learned a lot. I hope you did as well. As always, if you like what we do, please leave us a review on iTunes. Helps us in the rankings, helps us reach more people.
Thanks for listening to Prospective Doctors MCAT Podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school, check out medschoolcoach.com.